So I'm still trying to get a few things sorted out here. Okay. Let me quickly check. Okay. Just give me a second. Hope that this starts to work. I need to simply see if everything is already running or not. Okay, hello. I don't know. Are you able to hear me? It seems... yeah, now it seems to work. Okay, great. Usually always takes a minute or so until I'm able to actually also hear myself. Hi, hello and welcome. Microbehunter here. And after a couple of technical housekeeping issues, we're again ready to go with another microscopy, Saturday microscopy evening. Well, at least it's evening where I am. You can already see there, I found a flatworm in one of my water samples. As a matter of fact, there are lots of flatworms. I just lost it again. Yeah, I've collected a couple of water samples and I would like to simply explore them together with you. See, the flatworms, here it is again. Here it is. Okay, here we go. Yeah, I prepared this one first because it was kind of difficult to actually get out of my little plastic container because it liked to stick itself and adhere itself to the sides of the container. Yeah, but I think yeah, everything seems to be working. I see that several of you have already joined in into the live stream. Yeah, hello all around the world as always. And if you're new to this format, I just would like to explain to you a little bit how this works. Of course, you're welcome to type any questions and comments into the chat on the side. And after approximately 1 hour, I'm then going to answer your questions. I'm going to simply first do the observation session. Just want to show you a couple of things that I found under the microscope. And then I would like to answer some questions if you want to. Okay, so please feel free to type in questions into the chat. Yeah, we're basically ready to go. The live streams on Saturdays are usually quite improvised. This basically means that yeah, sometimes I don't even know what to expect myself. So what I've got here is I collected some moss from a pond. And I've got, well, actually I count this as one of the water samples because the moss is yeah, of course, surrounded by water. And I've got 4 other samples over here. And some of them actually prove to be what I think quite interesting. I collected this from different places, from the pond. Yeah, some algae, of course, some sediment from the bottom. And I found plenty of organisms in here, also plenty of segmented worms, flatworms, ciliates, of course, and so on. And every time when I pick out a sample here, I find different things. So I would simply like to observe this with you. Over the last couple of live streams, we've looked at prepared slides and permanent slides and did some other things. And today I decided again to look at a couple of living things here. Okay. Let me quickly check again a little bit. Yeah, there are a whole bunch of people have already joined in. Yeah, there are 43 people right now watching. Okay, so this seems to be working. Okay, yeah. So let's go back here again. So what I'm going to do now is I'm simply going to start exploring some of these things here. And if you yourself know a little bit, have something to say here to some of the specimens that we hope to find, yeah, then please, of course, also feel free to post comments here. Yeah, so what I'm going to do first, and again, I've only looked at a few samples here, but for example, this one over here I did not look at yet. That's some moss and some lichens that I'm isolated from the side of the pond. No, I'm not going to do that yet. I'm first going to show you a very short video, simply from the place where I picked the sample. I hope that this works. Does this work? Unfortunately, let me quickly see if I can get this. Unfortunately, I cannot get this to work. Let me see. Yeah, no, that's the wrong one. That's not the one where I actually picked it from. So yeah, I think somehow I had made a little mistake copying the specimen. Doesn't matter. In any case, I isolated. Let me put those tubes away here because I want to get a little more space. And generally those moss samples here, I do not have very high expectations, okay? And the reason is because the lichens that you see over here and the moss has been quite dry. I added a few hours ago, I added some water to allow the microorganisms, which hopefully are present, to kind of regenerate a little bit. But generally I found out that moss that is being exposed to dryness very frequently is not really a very good source for interesting microbes. But I just wanted to try it out in any case, and for this reason, I picked up some of the samples here. And usually, what I do is I apply a little bit here. Occasionally, I have also found some tardigrades, water bears, and so on. But this depends very much on the actual sample. So it can be that some samples are extremely rich in organisms and others are not at all. Here, I do not have very high expectations, as I already mentioned. But the reason why I still like to put them under the microscope is because this helps me also to assess a little bit in which places I'm able to find interesting microorganisms and where not. Okay, so even if we do not find anything, then this also gives me a little bit of some interesting, some interesting information here. So I'm just going to put it here, and I'm going to now place it here in my microscope again. Okay, and we're going to have a look. And this is basically what we're able to see here. And if you look very carefully, there are a couple of tiny dots whizzing around, right? And so there is actually microbial life present, but it's rather small. But then again, I'm also using a fairly low magnification here. Yeah, some green things that you see here might be some algae. No larger organisms here, but let's go up a little bit with the magnification. Put in the filter, everything here. I have to refocus again. Okay, and here we go. Okay, let's turn up the brightness a little bit. And so assessment, look, relatively small microorganisms, small is relative. Bacteria, of course, are significantly smaller. These seem to be either ciliates or flagellates, lots of debris. And I simply wanted to check this here. It confirms a little bit of what I expected, and that is that this moss samples that have been dry, exposed to dryness, usually are not quite as interesting as those that are growing in areas where there is permanent moisture present. So I'm just going to leave it at this here. I just wanted to have a little check. But I think the other specimens that I have are significantly richer in biodiversity. Okay. Here, the movement here seems to be due to surface tension of the evaporating water. This appears to be a little bit like a nematode worm here. This is most certainly a nematode worm. It's not moving. So. Okay. So what I'm going to quickly every now and then have a quick look also at the comments. But then again, I would like to answer any comments or questions which you might have later on. Okay. So hello all around the world, from Texas, the UK, from Spain. Okay, Frankfurt in Germany. So, Hydra. I just see here a comment, the last comment. Excited to see if you find a Hydra. Honestly, I've a couple of, I don't know, it's already maybe 1 or 2 years ago, in a water sample, I found Hydras. Here, the quick look that I had here, I did not find any Hydras. Okay, so it doesn't mean that they're non-present. So maybe we're lucky today. That would be really the highlight, but I'm not sure. Okay, so let's go and let's have a real look. Let's have a real look back at some of the samples here. Okay, and I would like to prepare now a couple of slides, and some of them I really did not look at yet. Okay, so we've already had a look at our little flatworm here, but over here in this tube here, I mean I numbered them here, right? There are some solid material here which settled on the bottom of the tube, and this is actually the thing that I would like to put under the microscope. Usually there is lots of stuff present there, hopefully, and in order to do that, I'm going to take a pipette. And I'm going to simply do the following. The pipette has a very, how do you say, narrow opening here. So this means larger particles might be difficult to be taken up. So that's why sometimes I cut it a little bit wider here. So I go all the way down into the sediment. And I agitate this a little bit out. There seems to be lots of sand and so on. I allow the sand to sink down a little bit. I apply a small drop here, okay, and the rest of the water goes back into the tube. And I will put a cover glass on top here. Now the thing with the sand is the following, that the sand grains are relatively tall, so this means the cover glass will not be completely flat on the slide, but that's okay. So you see that there is, it's not complete, it's kind of slanted a little bit, but for my purposes this is okay. And we're going to have a quick look. Again, under the microscope. Again, if you just joined in, please feel free to post comments and questions later on. I will try to address them. So, and this is, I have to go down a little bit more with the. So and that is basically the sediment from the bottom of the pond. There seems to be some kind of an ostracod, water crustacean, and as a matter of fact, a couple of days ago I was lucky. I was actually even able to find a small clam. Yeah. You know what? We're just going to have a look at this one over here in more detail. See, some of my live streams are simply just like today's, just simple observation. In other live streams, I talk a little bit more about theory. So I try to make a healthy mix. So that's very clearly looks like an ostracod, which are related to the no, it's not an ostracod. Seems to be some other kind of water flea. Yeah. In any case, either called water fleas or simply they're crustaceans related to the shrimp or the crabs, and we can see that they like to also be here in between the sand grains because this is probably also a place where they find some food. So let's have a look. Yeah. So let me have a look. I was actually quite lucky a couple of days ago to have seen a clam. And unfortunately not today, but you know what? I'm going to go down with the magnification again. Let's do it like this. Let's remove the filter because this gives me the best general overview. There is something in here. What could that be? Now it's a little difficult to see here because of so many sand grains. It might be some kind of a worm because it's kind of elongated. This looks like an oligochaete worm. Probably. Oh, that's Ialosoma. It's called. That's a classic. I've already made several videos over these. These are basically they belong to the segmented worms. Very common water organism. Usually lives between plants, eats debris, bacteria, and so on. Difficult to see now. Maybe we're going to see a better one later, but this one is now hiding between the sand grains. But my hope to find a tiny little clam so far has not yet materialized. Okay, again, one of those water crustaceans. In any case, I've been already filming also a little bit and recording the videos. So I would like to, of course, make a separate video, a slightly more condensed video of what I found here. Yeah. Again, a water flea. Pond water is, yeah, generally, not surprisingly, I think much richer in different life forms than, for example, when you keep some pond water in a jar. The reason is that usually when you keep it at home in a jar, what you see is some kind of a progression of organisms. It means some yeah, of them are going to really increase in numbers and others are going to disappear. Yeah. A tiny clam is a comment. Never heard of this. Please find one. Well, I hope I'm going to try again. When I tried this 2 days ago, actually I did find right on the first sample one, and I did make a video of it. So, but because I don't have one over here, I'm going to do the following. I'm just going to try it again. Okay, and just a second. Can you? Yeah, where is the sample again here? And let's try to pick up another one. And again. Okay. I mean, I said tiny clam. It was actually quite surprisingly large for. But let's give it another try here. And scope view. Yep, you see that there are others. Yeah, again, water crustaceans. There is lots of movement going on here, but there's so many sand grains. It's kind of difficult to actually see what they are. Yeah, it's again a worm. That's the one worm from before, the Iolosoma worm. 2 of them. Okay, you see some empty, what seem to be shells. Look at this. This is another one. Yeah, tiny crustacean. Yeah, again, some water fleas down here. This one is not a clam. That's this seems to be now an ostracod. Yeah. And this is basically what I think microscopy as a hobby is actually also about. It's a little bit the exploration. Yeah, and every time, even if you put the same sample under the microscope, you see different things, and that's essentially where you do have a little bit of a sense of I don't know, discovery maybe. Yeah, trying to find things that you have not seen before. Yeah. Yeah. Okay, there's somebody who's just been asking what's happening. Just came here. Well, I've collected a couple of pond water samples, and I'm simply putting them under the microscope, and I'm just looking at them and hoping to find something interesting. And right now, I'm looking a little bit for a clam. And you know what I'm going to do? I don't know if this actually works. Just a second. Let me try. Let me try. While you're looking at this here, I'm going to try to find something else. Yeah. Maybe this one here. You know what I'm going to do? This is not, forget about this video, forget about this. I'm going to try to change this video around. I'm going to try to change this video because if we cannot find a clam, then maybe I'm able to at least show you a video of one. Okay, so that's the recording that I made yeah, just a couple of days ago. So that's not live right now, but that's actually a video recording. Okay, and this is a clam that I found in the very same sample, and I basically isolated it. So I removed this, picked it out from the sand, and basically I just wanted to yeah, have a better look at it, and this is how it looks like. Yeah. So yeah, so this is basically exactly from exactly the same sample. Okay, and then I took my pipette and I took it off the slide and put it into some clean yeah, area of the slide without sand grains, and this is basically how it looks like. And I thought this was kind of cute and some kind of cool. And if you look carefully, and I think I've heard this somewhere. If you look carefully, sometimes on the shell you see those rings, almost like tree rings. And based on the number of rings, you can actually determine how old the clam is, the age of the clam. Look, I think it's kind of okay. Now the video is looping. Okay, but what I would like to do is actually it's the same video now, but I actually could show you also maybe a slightly different video here that I also recorded. Yeah, that's another recording that I made the other day, with a higher magnification. Yeah, and yeah, it's quite nice. Yeah. Yeah, so this is yeah, quite nice. So basically what happens is those clams will actually grow in size, of course. And I know that in this particular lake and pond, there are a lot of clam shells and clams there. And they're about this size, you know. But I kind of like this one here because it's actually a microscopic version. You can see that a large part of the clam is actually a little bit blurry as well. That is because it is fairly large. I mean, maybe, I don't know, maybe a millimeter or something, right? And for this reason, some of the shell and the clam is a little bit blurry and out of focus because of the limited depth of field. Okay. Yeah. So this is, yeah, quite a nice thing here. By the way, this is not in time lapse. It's just regular. Yeah, regular speed. Yeah? And another thing that I kind of noticed with many of those tiny animals is look here on the side, all the way on the left, that's the edge of the water droplet. Yeah? So it didn't go out of the water droplet. And so that's also kind of interesting. It's, someone maybe is able to sense that this is maybe the limit of the droplet and it did not go out. Yeah? So it liked to stay in the water here. Yeah? And the nice thing here is also because of the small size, the shell is also a little bit transparent, and therefore you're yeah. Ah, yeah, the shell is a little bit transparent, and that is because of the small size, and therefore we're able to see some of the structural details. Like, you know what? It is because it's a video. Let me quickly pause it. Yeah. So yeah. So now the video is paused, and I think now we can see much better. Yeah, the structural details and. I don't know. I'm not an expert in mollusks, but apparently the growth. You can determine somehow the age of those clams by also counting those rings here, because every year apparently, almost like in a tree, yeah, those rings are deposited because those clams also grow, of course, primarily during the time when it's warmer. Yeah, during summer, and therefore every year somehow a ring is deposited. I heard somewhere. Okay, so but I don't know. Let's go back again to the sample. Where is the scope view? Here we go again. Okay, so that's not a live. That's a live picture again from a microscope. Yeah, if you want to have color, I can add the prism here. Yeah, let's have some color as well. Doesn't really make a huge difference. Well. Yeah, it's a little too dark, I think, so maybe we're going to open up the condenser a little bit. But sometimes it's going to be a little bit overexposed. Yeah. You know what? I'll give it a last try. Yeah, but now that we've seen, this is kind of shaking. Let's give it a last try. I'm going to take another sample now from the tube. And add it on here. This, by the way, is without cover glass. Okay, so yeah, so you see that I'm using the lowest magnification here, and therefore the distance between the objective and the sample is relatively large, so there is no danger. And I'm going to just pick another one here. And where is the desk view? Here we go again, and here it is. And let's try another one. So that's, by the way, sometimes people are asking me, well, I put water samples under the microscope and I didn't see anything. It's because you do not put the actual water under the microscope. There is too little material in here, right? Unless the water is very green and cloudy, well, then you can do that. But this water is fairly clear, so you will not find that many interesting organisms in there. So you want to go down here into the sediment a little bit, yeah, and yeah. I'm going to now spread it apart a little bit more. Yeah. And let's give it a try. So I'm adding now the second slide here. Okay. It's a little bit out of focus again. It could, of course, also be that maybe some of those clams, maybe they actually went further down into the sediment. See, there's less. Now, all of a sudden, there is less movement here. Yeah. So maybe I've taken something from the. You know what? This is almost. You see how different it is. It's almost no movement. So maybe because I took something from the surface, could that be the reason? Maybe the organisms also all moved further down to the bottom, maybe? Because the difference is quite remarkable. Compare this here to the one from before. Yeah? Well, was much more movement going on here. Yeah? So that's the one from before, and this one over here. No movement. So you know what? That's my hypothesis. So my hypothesis is that the organisms, whatever they might be, kind of moved further down. So what I'm going to try now is I'm going to just try to take a sample further down from the sand, from the tube. Okay, so I'm just going to use the same slide again. Where is the desk? Here we go again. And that is actually an interesting. You see the tube is conical, so basically it goes together to yeah. Yeah, so I'm going to try to reach in and maybe get something from the very bottom here. Not knowing if this could be the reason, but let's try it in any case. I'm going all the way down. And I've taken a sample, actually quite a lot, all the way from the bottom. This is very dense. Maybe a little too dense, but at least we're able to see then whether there's something in here. You know what? Maybe you can spread it apart a little bit by tapping on it. So, I don't know. We're gonna give it a try. Scope view. So, and here it is. That is now the one sample from the bottom. Interesting. Huh. There's a little bit. Where have they all gone? Could this be that's a clam. I think it's a little bit smaller than the other one I had. But maybe that is one. Yes, it is. Well, it's blurry. Just a second. Let me focus. Here we go. Okay. Yeah. So that's again, yeah, a clam. This shell is even a little bit more transparent than the other one. I just realized. Yeah. And what you can do, I mean, I could try. You know what? I'm just going to demonstrate it. You know what? No, I'm going to demonstrate it a little bit later after we've watched a little bit, because there's no guarantee that it's actually going to work what I'm going to do. Okay, and then we're going to probably lose it again. So we're just going to watch it maybe for a minute or two, and then I'm going to try to use my pipette, and I'm going to try to pick it off, and I'm going to try to transfer it to a clean place on the slide. Yeah. So this is basically, I do not dare to go up with the magnification yet further, because again, there is no cover glass on here. And if it is now the 10 times objective and never go up to 20 times, then there is the danger, of course, that I'm going to touch the water with my objective, and I don't want that. Yeah. So I'm just going to then demonstrate to you how I pick it off, and if it doesn't work, then at least you know what I've done. Okay. Sometimes there is a little bit of trial and error necessary. And you know what? While you do that, while you watch this, I'm just going to quickly reach over. Just a second. You can continue to watch this. Just needed to get a lighter. I'm going to show you why I'm using a lighter as well. Okay. Yeah, let's admire it for another minute or so. You can actually see how that is actually opening up the two, yeah, the two shells. Yeah. Yeah. So it's actually quite nice. Just a little bit of some background information about the pond where I collected this. This was actually a pond with very clean water for swimming. So I spent there a day a few days ago. We have a very warm summer, so we spent several times. We visited the pond or the lake rather. Yeah. Yeah, so I'm going to now show you how a possible method on how to isolate it. Okay, so this takes a lot of patience. Yeah, and maybe it's not going to work because maybe I'm just shaking a little too much, and yeah. So this could indeed be the case, right? But if it doesn't work, then it doesn't work. Yeah? So we're not going to feel upset about it, but I just wanted to demonstrate this to you. So this is basically where we've got the clam, and I'm going to just quickly show you again the desk view because I need to do a little preparation. What I got here is a lighter, and over here I've got some pipette tips. You've seen them, they look like this. They're disposable plastic tips, and they go on here on my microliter pipette, and I'm going to use a new one that's not wet. I'm going to trash this. And what I would like to do is I would like to go into the water with this pipette on the slide and take out the clam. And in order to do that, the clam is probably bigger than this one over here, right? So what I have to do is I have to probably cut away the tip again. So to make the opening large enough for taking up the clam. But then there is now the problem that if I go on the slide at an angle like this, then half of the opening is going to be open and I'm not able to pull up any water, right? Because air is going to go in. So what I have to do is I have to try to bend it down a little bit because I cannot hold the pipette steeply with a steep angle. I cannot do that because there's no space. So what I want to do is I want to hold the pipette flat, but the tip should be bent downwards. Okay, so it's easier for me to take up the specimen. So what I usually do is the following. This is a little dangerous. I need to go on. I've got a camera here on top, so I better not burn the camera. And I quickly warm it up, and I bend it down like this. Okay? And I wait for it to cool, and we're finished. Okay, so like if I basically have now, it's much easier for me to pick up the specimens. Okay, that's it. Yeah. So it's already cold again. Yeah. And yeah, this is how it looks like. Yeah. And I hope to be up. No, it fell off. So basically, I kind of hope to go over. I just demonstrate this to you now. I'm just going to take a blank microscope slide here. So basically, I just hope to kind of be able to pick it up like this, and then I want to transfer it somewhere else. That's the idea. Yeah. So let's try this. So I need again the microscope view. I have to find the clam again, which I lost because it's been going somewhere. Up, some movement. Here it is. Here it is. Do you see this? Okay. I think this seems to be it. Let me go up a little bit higher. So yes, that's the clam. Okay. So that's the little fella that I would like to isolate. So I'm switching over now to the lowest magnification. Okay, it's always have to readjust. So, and let's give it a try. Okay. So I'm going to disappear a little bit out of the. I'm going to disappear now out of. You're not going to see me. You're just going to see my green wall in the background. But maybe you want to look both at the top. Okay, because this is where you're able to see what I'm doing, and also on the other side over there, where you actually see the magnified image. I need to pull over if I want to talk. I need to move the microphone over here as well, and let's see if this actually works better like this. Okay. So let's give it a try now, and I somehow will try to I will try to do this looking through the microscope. Okay, so you apologize that I'm kind of disappearing right now. I need to find the clam again over here. So here we are. See, it's a little difficult to see, and I'm kind of holding. Look, I'm kind of supporting it like this, right? So. I don't know if I got it. Maybe I did. So let's move over to a clean place of the slide. And let's see if it was successful. Nope. It wasn't. Could there be maybe something still left in here? Nope. So maybe it's still there somewhere. I don't know. You see, that's the thing that I was kind of worried about. It's still there. Here it is. I couldn't get it. Okay. So let's give it another try. That's what I said is you need quite a bit of patience with this. So I usually go all the way in. I rest the pipette on the glass and chuk. I hope that I got it. Let me check here if it's not here anymore. Chances are pretty good that I actually got it. Or is it over here? Ah, it's still here. Here it is. Okay, let's give it another try. So, maybe I need a little more water. Here it is. So if this didn't work, then I don't know. So let's give it a try here. And here it is, the clam. Okay, now you're gonna say, yeah, it's not moving for whatever reason. Ah, it is. It's moving. Here it is now in the center. Now you're gonna say, well, there is still quite a bit of sediment here, sand grains and so on. That's correct. So what I usually do is the following: is I add more water. Okay. I add more water. And I try to repeat the whole thing. Okay, here it is. Usually after it's agitated and if there was movement, then it stops moving. Okay, and then I usually add water again, and I pick it up and I repeat this several times until all of the sand is gone. Okay, and this is how I actually got this nicely isolated clam, without any other material. Yeah? So it's kind of by repeating, with a lot of patience, by repeating, picking it up and adding a little bit of water, and I'm able to kind of isolate the specimen that I would like to have. Yeah. So somebody just said is that there are two of them. This could be, I don't know. That's quite well possible. Yeah. So So this is a kind of the way that I think it's kind of really interesting. So Yeah, so that's I just wanted to show this to you. Okay, I'm just checking a little bit. 39 minutes. Yeah, and we're just at water sample number 1. Yeah. If we want to grow them, what would they eat? Generally, difficult to say because the natural environment is complex, but organic material. So if you have got some algae in there, right, and usually they like to, because they're on the surface, they like to eat the stuff that's kind of growing on the biofilm on the surface, right? So yeah, just like snails, or the filter feeding, that means they're kind of filtering the water. So usually debris and so on. Honestly, I've had mixed results. Sometimes when I feed my water fleas, I add some yeast, right? But that's suspended in the water, so of course water fleas like to eat that. But those clams, they eat like many other organisms, suspended organic material. So if you've got some decomposing plant matter in there, biofilm, then basically they will eat that. Yeah. However, I think it's not only a question of feeding, because sometimes, if the temperature is not correct, if the oxygen concentration is not correct, you see, then they're also not going to grow, right? So there are so many factors and variables sometimes that essentially. Oh, thank you! I just received another donation. Thank you very much. Okay, for your info, with the donations I'm using to again buy stuff for microscopy that I can show you. Okay, for example, some slides and whatever. Okay, so we spent quite a bit of time now with this clam here, and usually, honestly, I know this might. I'll be honest with you. You know what? I could simply wait and take the slide and rinse it and wash the slide, right? Usually, I have a difficult time doing that. Especially after I've looked at the microbes, and the clam is not a microorganism, but still, after I've looked at them for such a long time, it kind of sounds weird if I say it, I kind of feel a little sorry just kind of washing it and kind of removing it that way. So usually what I do is the following: I put it back into the container, okay? So it might sound a little bit ridiculous, okay, but yeah, why not? It's not going to survive forever in the tube anyway. I know about that, right? But still, yeah, just for the sake of being nice. And you see, I'm not able to take it up, and the reason is because there's too little water there, so there is still a little bit of air going in here. So what I could do is I could add more water. So I'm going to add a little bit more water, and then I'm going to withdraw it immediately. Okay, let's just try this. Thank you very much again. I received another donation. So a little bit of water, and I've sucked it in. I've sucked it in now, okay? So it is in here somewhere, and now I can put it back into the tube. I usually rinse it out, yeah, and that's it. Okay, so that's kind of the thing. Usually, another comment. I know that some of you have joined my live stream already many times. It might sound a little bit repetitive in what I'm doing, but some of you are new. So for this reason, I'm going to also give you a couple of tips here. Those plastic containers are, I think, quite useful for storing water samples. However, I do recommend that if you collect water, always make sure that there is at least half of the tube contains air. Okay, and then it's fine if you put a cap on top. And then usually I open it once a day to allow fresh air. I usually go like this to allow fresh air to go in. And then you can actually store it for a fairly long time. But, and that's important, if you fill up the container with water and put a cap on it, then it's not going to store for a very long time, because the whole sample tube is going to become anaerobic. It's going to start to rot and decompose, and it's going to start to smell foul after a few days. And you don't want that. Thank you very much. This was a very generous donation from Scott. Okay, thank you very much. And it's going to start to smell foul, okay? If you fill it up after a couple of days. So my recommendation is to only use a small amount of water and sediment. I'm telling you this because a couple of years ago, when I've actually collected the samples using those tubes for the first time, I was visiting another country. I wanted to take some water samples back, and I filled it up because I wanted to take along as much as possible. Filled up the tubes all the way. And then over the course of a week, it started to actually smell bad, right? It went anaerobic, and then the organisms started to die. And there was one tube, however, which contained only very little water, and it was absolutely okay. Yeah. So over here, for example, this one is already also a little bit more on the generous side already, right? So what I'm going to do now is this was the sand sample. Okay, I'm now going to pick up a little bit of this, this material that I've got over here. Okay, and let's have a look here. I know that there are also some, you see those dots in here? Water fleas, water crustaceans swimming around. We've seen those already. The greenish stuff here is some algae that started to grow already on the inside of the container. There are some flatworms here, which I already showed you one at the very beginning, because they're really difficult to take out because they like to stick to the side. But let's have a look at what we find here on the bottom. Yeah? So again, I need to do the following here. Okay, it's kind of sticking together. I've actually been thinking also about a little bit, and that's also maybe something I would like to talk about. Normally I'm talking now about the samples and so on, but maybe I would like to talk also a little bit about some experiences that I have had as a biology teacher. That's the stuff that I'm doing in my real life, in my non-YouTube life. I'm teaching biology, and school's starting again in 2 days on Monday. So after, the new school year is starting, and I'll be teaching again a new course in biology. So let's look at this, all of this stuff. Yeah, this is a general rule. If you are able to see it with your without a microscope, like the stuff over here, then generally you're also going to see something with a microscope. Yeah, but a clear water sample, it's a little bit out of focus. I'm just seeing a clear water sample sometimes does not contain a lot. So let me take again. In this case, I'm going to take a cover glass. Let me see which one. Those cover glasses I washed, therefore they're not, and they're reasonably clean. And let's go like this. It's a little bit more on the generous side, but it's okay. Yeah. So let's have a look. I need to remove the other slides first. So 1 and 2, and let's put it in. I have to change the camera. The scope. Here we go. Where is it? So here we go. Okay, and not a lot of movement, but other interesting stuff. Okay, see that it's the same pond, just taken from different areas. So you see the one before, we had a lot of water crustaceans and so on. So let's go in a little bit further with the magnification. So let me see, just a second. Here we go. Yeah, and that is essentially, that's the stuff that the clams and the other organisms like to eat. Lots of algae, lots of decomposing stuff. But the thing that I kind of really am looking for is we see some diatom shells here. And that's actually another thing that I once, a couple of years ago, tried to prepare to make diatom preparations. So here we go, a little bit more light. Yep. Yep. Some bacteria, the small ones over here. The sample is fairly thick because I used a lot of water. Yeah, so those things here, let me point again. Where's the, here's the arrow. Yeah, these are of course diatom shells made of silica. Also these here, this one over here. They do photosynthesis, they're pigmented, therefore, however, there is a protocol in order how you are able to isolate and extract those. So I'm just going to tell you how this would work. I've tried it a few years ago, but have been a little bit hesitant about doing this because it's a little bit dangerous, the method. So how would you purify those diatom shells? What you see in here is you see a lot of other algae as well. However, those diatoms over here, they have a shell made of silica, which is chemically similar to glass. So wouldn't it be kind of nice to have a method to kind of remove everything else except those silica shells? And there is a method, and that would be is by adding a quite strong hydrogen peroxide solution. Hydrogen peroxide is a very aggressive oxidizing substance, and you add around, I don't know, 30% hydrogen peroxide. That's not even easy to get because it's so aggressive. Normally you use 3% hydrogen peroxide for disinfecting. It's also be found in hand sanitizers, but you need a fairly concentrated solution. And when you add that to a sample like this, it's going to remove everything that's organic. It's going to oxidize it away and convert it to CO2. So all of the organic stuff that you see here is going to be removed, and the silica itself is not organic. Yeah. Although the contents of the diatoms are going to be removed. So there is a possibility to to really isolate those shells. For example, here is a very nice one. But again, I've been a little bit hesitant of doing that, because if you get anything of this concentrated H2O2 hydrogen peroxide solution into your eyes, you're in real trouble. So you need safety goggles and so on. Yeah, but that would be it's not a method that I invented, I researched it. That would be a possibility, right? In this case, for example, by adding hydrogen peroxide, you would also remove the green chlorophyll inside the diatoms, yeah, and you would get the very nice diatom shells. I've also read another method somewhere where people suggested, and however, apparently it doesn't work quite as well, that you dry a sample like this and then you try to heat it up to a very high degree so that everything else burns away except the silica. But there is usually also some charring and some soot left over, so it's not quite as nice as using hydrogen peroxide. Yeah? So I just wanted to share. Pediastrum, it's called. It's a colonial algae. Let's go up a little bit with the magnification. Let's go up here. Yeah. Yeah. So I need to maybe go a little bit like this. That's the name. It's a little dark. You know what I'm going to do is I'm going to change now the camera. I've seen there's been a question about my camera, so I'm going to answer this as well. That's a colonial looks like almost like Pediastrum, which is a colonial algae. Yes, it is. Yeah, you can see the individual cells. You can see the individual cells here. Yeah, and those spikes on the going to the outside. Yeah. So let me see, what else do we have here? Yeah, again, those diatoms. Here's another nice one. Yeah. So the thing is the following. If I'm reading, of course, also microscopy forums and so on, and some people are saying, well, the problem is it's all quite nice and interesting, but the problem is that you always keep on seeing the same things. It's always algae and diatoms, and of course, the first time when you're seeing some of these things, it's all interesting and exciting. But then if you see it a 10th or 100th time, yeah, it's kind of becoming a little boring. I understand that. And for this reason, some people are saying I'm starting to lose a little bit the motivation because it's always kind of repetitive. And I'm saying, well, not necessarily. What you have to do is you have to keep yourself motivated by digging in deeper. You might think, yeah, of course, these are just diatoms, but which ones are these? Okay, so get yourself some identification books. I don't even know the names of them, right? In a live stream, I don't have the time to actually search now. It would be also kind of boring for the viewers to kind of search now for all of these things in identification books and so on. But that's what my suggestion would be, is if you just basically scan those samples on the surface without digging in deeper, then yeah, it can indeed become repetitive. right? Everything is going to become repetitive, obviously. But actually, if you now kind of try new extraction techniques and play around a little bit with different filters and so on, then there are so many things that where you can actually keep yourself motivated and add variation to the hobby. Yeah. So I'm always checking in a little bit the thing here. Yeah. I'm going to, you know what, because I do want to check the other samples as well, okay? Because I have not checked all of them yet. So this was a little bit one from the very bottom. So this is, I'm also going to move this to the side. So the desk view here again. So that's the sample here. I'm going to put this to the side. I've got two more. Again, different forms of algae. And look, this was a water plant that was floating around. Let me try this one here first. The thing is the following. In many cases, water plants, I think it might be it's a very common plant that's also found sometimes in aquariums. Sometimes there is stuff growing on them, on the plant itself, and that's actually what I'm interested in. You see that it's maybe not completely submerged in water. I'm just going to go in, going to take out, hopefully, being able to take out a small part here. Let's use scissors maybe to, and I don't know how interesting or not interesting this one is going to be. This one is pretty thick, obviously. So let me put this back in. Okay, let's put everything completely in. I need a bit more. Up. Wow. Oh, gee. Did I break my... I just broke my pipette. That's no, I didn't break it really. The front part here came off. Okay, so I have to put it back in again. Okay, yeah. This mechanism here is for ejecting the tip. And I just kind of thrown off everything here. So, but now it's working again. I'm going to add a bit more water. And this is extremely thick, okay? So this is, that's actually a little bit too much. Usually you want to separate this a little bit, and let's put this back. And let's have a look. First, you know what, I'm just going to add a cover glass just to be safe. It's very thick, but yeah, let's see, okay? Maybe we're able to find something interesting, maybe not. That's again always the kind of the surprise aspect here. Oh yeah. Yeah, already at first sight, I'm not seeing a lot of stuff here growing on the surface. Here, at least we got that guy. Ialosoma, that's one again of one of those worms that we've got here. I'm hearing something again. Thank you very much. Yeah, your questions just arrived here. So, and yeah. Okay, so usually on the surface, or often on the surface a little bit, we're able to see here. Sometimes on the surface, especially if the water plant is a little bit older, you're able to see some either some cyanobacteria or diatoms or whatever growing in a thin layer of biofilm here on the surface. I don't see that so much here. You mean maybe this one over here? Sometimes Vorticella and other interesting, more interesting organisms, but not so many here. What is this long thing? Ah, yeah, that's an algae also. Yeah. So, yeah. So, many of the higher algae, like we've got over here, you notice that they're not green, but more brownish in color. That is because the further deeper down in the water, there is essentially more blue light is able to reach the deeper parts of the water, and therefore you need a reddish brown pigment in order to absorb those wavelengths better. So there's a well, I don't know if this is not a little too specific, but essentially the color of light that those water plants obtain further down is different than at the surface. Yeah. So for this reason, water plants that are growing deeper down actually have a more brownish reddish color because this is they're adapted this way to absorb those wavelengths better. Yeah, so okay, yeah. So this is basically how it looks like. We can see that there are, it's not so much growing on the water plant. So maybe I think it might have been a little bit too young or too fresh. Yeah. If it's the more decomposing ones, there is more living on those. Yeah. So this is the thing. I'm going to now. Yeah. Occasionally, we're not going to do that now, but you see over here this round thing in the center, that's an air bubble or oxygen bubble. Sometimes it will actually grow with time. Yeah, and that is because the plants or the algae or whatever, they are starting to produce oxygen. So if you see bubbles like this, then yeah, it could be actually a sign of photosynthesis. But sometimes, depending a little bit on how much plant material there is on the slide, the growth can be fairly, of those air oxygen bubbles can be fast or slow. Yeah. Yeah. So also not. Yeah, I'm just going to move on now because I do have the last sample that I would like to show you before I move over to the questions that you have. So, and I'm going to take another slide here because. Yeah, so let's have a look here. And here I'm just, you cannot see this because, here we go. Okay, so now I'm reaching in with my tweezers, pulling out some of this solid material here as well, which is kind of difficult to get. Let's try it like this. It's the same water, so I'm just going to use the same pipette. It's just taken from slightly different places of the same pond. And let's give this one here a try. Scope view. And yeah. You see? Again, some different characteristics. Spirogyra. I always have a problem finding out the proper pronunciation. These are very common algae, which have a spiral-shaped chloroplast. Let me show this to you. Let's open this up here a little bit more. Let's remove. Let's go like this. Let's go up yet one further. Up. I bumped it out here. Yeah. So those, yeah, it's very typical. A very common algae, and what they have is, again, I'm going to turn on the arrow. Yeah, you see that over here, the chloroplast, which is responsible for photosynthesis, is kind of spiraling inside the cell. Yeah, it's quite, it's quite a typical characteristic. Yeah. So. Yep. Yeah, so let's go down a little bit with the magnification again. Let's put this here up here. Let's like this. Yeah, now we're able to see it a bit better. Here we go. Forgot to put in the condenser. So this is a little too high again, because to get a better overview, you always want to use the lower magnifications. So yeah. Let me see. Yeah. What is this here? Vorticella. Okay. That's a ciliate. Okay. Some traditionally called belled animalcules because of the bell shape. Yeah. And yeah, what they have is they have this one cell, and they have basically this long extension here with which they attach, and usually when disturbed, then they're able to retract very quickly. And there's usually a ring of cilia, of tiny little hair around it, which generates a water vortex. So sometimes you're able to see the water streaming around, at least the particles that are in the water stream around. So that's Vorticella. Here again a little bit. Yeah. Yeah. I don't know if maybe what I'm gonna do is I'm gonna try to tap the microscope slide. No, sometimes some of them will actually retract. Yep. Now, do you see that? Yeah. The stalk spirals together and it quickly retracts. Yeah. So let's try it again. I'm lightly tapping the cover glass. Yeah. Okay. And spirals together. It's actually what I read somewhere is one of the fastest reactions or responses known in the living world. It's the speed with which it's able to retract. Yeah. So yeah. So let's have a look. Okay, so what time is it? It's already 1 hour, and what I'm going to do now is I've been through all of the samples anyway, at least a very quick look. If you spend more time, and of course you're probably going to find more, more things to observe. So it's a question of patience. Here again, our little Iolosoma worm. I've been playing around with those guys a lot. If you chop them in half, then you've got 2 of them, and both are able to survive. Yeah. And if you look at the head, for example, then you see that maybe those tiny, some of them are red dots. These are oil droplets. So it's oil droplets or lipid droplets. That's why it's also known as the oil droplet worm. Yeah. And what it does is it reproduces asexually, among others. So that means it's able to actually divide in half. And when it divides in half, it's going to separate, and then there are 2 of them. So those guys have also a very remarkable ability to regenerate. Yeah, so you know what, folks? I'm going to now move on. I'm going to move on now, and I'm going to have a look at your questions, and I hope that I'm able to answer some of them to your satisfaction. Again, I've not read them yet, so I don't know exactly yet what you're going to ask. Okay, so here we go again. I would like to thank again Michael, who put the questions together. Okay, and let's go through them here. Do the tardigrades that are the only species to put themselves into anhydrobiosis? I've noticed rotifers rehydrate on moss as well. Is this the same mechanism? No. So first of all, anhydrobiosis basically means that tardigrades have a remarkable ability to withstand dryness. Apparently not high temperatures, what I heard, but dryness and some other organisms as well, for example, rotifers. I don't know if the mechanism is exactly the same, but I do know that different organisms do indeed have different mechanisms to withstand dryness. Even some bacteria, for example, bacillus, they are able to form so-called endospores, which are able to withstand dryness and which are able actually even hundreds of years. Right. So about the mechanisms, I don't know, but I just want to. That's what I do know is that indeed the formation of kind of cysts or other similar structures that were able to withstand for a longer time is something that can be found in biology quite well. Yeah. Did you deliberately not put a cover glass on? Yes. Well, that's correct. Sometimes I do not put a cover glass on. When? That's the question, okay? So only when I want to make a quick check, normally, I just want to see is something there or not. When I'm using low magnification, like the 4x objective. Why? Because with a low power objective, there is no danger of actually touching the liquid, the water. Right? I don't want that. And then sometimes when I want to actually manipulate the specimen, like for example, what I've done before, I've taken the pipette and I've taken some of the samples off, right? Of course, you cannot do that if there's a cover glass on top. Yeah. So it depends really on the situation. Generally speaking, for the low magnification, like the 4 times, it will not make a huge difference if there's a cover glass on it or not. The reason is that the depth of field is fairly large anyway, and you will not see a significant change in image quality. For the higher magnifications, a cover glass is very important, not only to improve the clarity, and to protect the objective from not becoming wet, but also because it limits the movement of the organisms, right? Because the cover glass kind of presses it flat. Otherwise, the organisms are going to move all the way up and down, and then you always lose focus, right? And that's actually can be a problem. Yeah. So the answer is yes. I deliberately did not put a cover glass on. Yeah? Does this calm baby one? So that's the clam, right? Supposed to become a big clam. So what or is it a microscopic species? Basically, does it stay small like this? Let's put it this way. I'm going to give you now an answer. I don't know the answer for this specific one, right? But I do know that big clams obviously have to start out as small ones, but I don't know if this specific one actually stays small or not. Okay, I don't know. However, I'm going to give you now an answer that my university professors have, or one of my thank you very much again for a generous donation. One of my university professors has given me, and it's a very useful general answer that I always tell my students when they ask me difficult questions where I do not know. And the answer is the following: if using common sense, with a common sense biological knowledge, is it conceivable that this could be a clam that stays small? Okay, it's possible. Why not? Then probably somewhere in nature you're going to find clams that will stay microscopic. Okay, as long as it doesn't violate any laws of nature, so to say, then it's possible. Yeah, it's possible, and that is I think a very useful answer. Generally, students sometimes ask me, "Is this possible?" And I say, "Well, I don't know, but do you think it could be possible?" And then the students say, "Yeah, actually, I think it could be possible." And I say, "Well, then probably if you look hard enough in nature, you're going to find an example where this is the case." Yeah? Because nature is simply so extremely diverse. Yeah? So that's essentially the answer here. I know I'm avoiding the answer. Is it going to stay small? I don't know. It could become big, and if you look hard enough in nature, probably there are going to be a whole bunch of microscopic clams as well. Yeah? I imagine the mangroves would have some interesting mix of land and sea critters. I have not tried it properly yet. Biodiversity is high in many natural areas. I would say I don't have mangroves where I'm living, right? But I would just have a look. Yeah, I've heard we can put vinegar on chalk and under a microscope, and we see microfossils. Have you ever tried? I get it. Okay. Makes sense. I need to explain this here a little. This does not work with commercial chalk that's artificially made. Okay, sometimes in teaching you use chalk, and this is usually some kind of a commercial chalk that's artificially made, very pure, often a mix of I think calcium carbonate and even calcium sulfate. They kind of mix it together to make a nice chalk for proper writing. But there is also natural chalk out there. Natural chalk contains calcium carbonate. And if you add vinegar or any other acid, then this calcium carbonate will be dissolved, okay, and will be removed. So I can indeed imagine that you're going to find some fossils in there, but you got to be careful because many of those fossils, like seashells and so on, are also made of calcium carbonate. Maybe they're a little bit harder, right? But you got to be careful that you're not using vinegar and that you are also removing those fossils. Okay, but I think this could be something. But do not try commercial chalk that you can buy for writing on chalkboards. You got to find some kind of a natural chalk, where you're able, which contains those fossils. When I'm trying to pick something off a crowded slide, I'll cut the tip of the transfer pipette so that it's beveled like a needle. Ah, I see. Okay, so this base, that's thank you. Okay, good point. I don't know why. Honestly, I didn't think of that. So I'm going to demonstrate this to you. So what I've done is I bent. I did it the complicated way. So I've basically used the lighter to bend the tip here, right? But why not? And this is the suggestion here. Why not cut it? Why not cut it diagonally? Okay, you have the same effect. Yeah, you can actually pick it up like this here. Thank you for the suggestion. Honestly, I didn't think of that. I don't know. I've been thinking too complicated. Yeah. So, how does the clam look like in dark field? I don't know. I'm now read it too late, and yeah, that's my mistake. So you know what? But thank you for the recommendation. So what I'm gonna try to do is because I want to make a separate video of those specimens anyway. I'm working on those. Then I can actually try to find a clam again, and I'm gonna put it in dark field. Okay. Yeah, I think it might actually look nice because it was semi-transparent. What would they eat? The clams, as I already mentioned before, sediment, usually sometimes also because they're kind of scratching along the surface, biofilm. Yeah. I don't know if it's so easily possible to feed them. They don't need a lot when they're that small. So I would say that if you take a natural water sample with some water plants and some debris in it, then you're going to have plenty to eat. The clam actually utilizes calcium in the water to build their shells, and the thing that moves the clam is known as the foot. Just like in mollusks, also because clams are related to snails, right? It's also referred to as the foot, the muscular foot. Speaking of changes in water samples in a jar, how is your mini aquarium you made several months ago doing? It's standing over there and it's not looking nice. I'm going to show you, just a second. Okay, here we go. I'm actually thinking about making maybe you can see it over here. The water plant, the Elodea, completely died and decomposed, right? The water fleas, the ostracods and so on, they survived. Yeah, there is now much more, I don't know if you're able to see it, but there's now much more stuff here on the bottom. There is now biofilm, so basically there's some algae now growing on the rocks here as well. So snails have grown here. I don't know if you're able to see this, right? So yeah, not optically not nice looking. Yeah, but still stuff to observe and put under the microscope. Yeah. So this is basically this mini aquarium, which I kind of kept going. I did not exchange the water, but I always kept on refilling the water when water evaporated. Yeah. I will tell you some additional information. What I tried to do the following is maybe this was a bad idea because this is when it really started to collapse. I wanted to feed the ciliates in there. It's possible to make a so-called a milk culture, and what you do is you add a tiny droplet of milk, a very small amount of milk, because the fat in the milk can also be used by microorganisms as a food source. And ever since I've done that, and then all of a sudden the population of water fleas completely collapsed. I don't know if it was only because of that, right? Or if some other factor contributed, but in any case, I stopped doing that, and I've been now adding again some yeast and to kind of regenerate this. Do you see the slide on the microscope on the screen with the broadcast software, or is it on the camera software? So what I've done is that's a technical question on how I have the whole thing set up. I've got up to a total of 6 cameras connected to my computer. And depending on the scenes, I'm using a program called OBS Studio. Depending on the scenes that I'm using, different cameras are basically put in place. So for example, right now, this one over here, that's a webcam on top of my monitor. This camera that films the sheet and desk, that is simply a desk camera. Yeah, you know what? I'm going to just lift it up into the other one here. That's how it looks like. It's a document camera. Yeah. And for the microscope, I've got a Canon camera, and this Canon camera has an HDMI out. And I now put this HDMI out into the computer. I've got a little adapter, so the HDMI is converted to USB. which then goes into the computer, which goes into this program here. It's also possible to use dedicated camera software. I've also done that before, but I decided simply to use HDMI. Yeah, so yeah, for ease of use. Yeah. Put an x-axis and y-axis on the bottom and left, and so that you can tell the coordinates of the thing that you're specifying. Yeah. Interesting. An x-axis and a y-axis on the bottom and the left, so that you can tell. That's a cool idea. So basically, what I've been doing up to this point is I've always used an arrow to kind of show around the thing. Look, yeah. So basically, like over here, it's a little bit out of focus. If we were already talking about the whole thing, let's at least put it into focus. I've got a little arrow over here, which I made. It's semi-transparent, which you can switch on and off. It's just a graphic that I overlaid, and I'm able to use this to point. But essentially, the suggestion is maybe why not have some kind of a coordinates here on the x and the y axis. So there where you can just say x10, y25, yeah, and then basically, yeah, you know exactly where I am. That's a suggestion. Never thought about that. Thank you. Okay. And for the Q&A, follow-up questions on the last week's discussion on hydrogen peroxide. Could I use hydrogen peroxide to isolate rotifers mass stacks by dissolving the body of a dead rotifer? Well, if even though I never tried it, if hydrogen peroxide will dissolve everything that is organic, okay? So if the mass stacks, and I don't know what the mass stacks is made of, if the mass stacks, that's this grinding organ in a rotifer, if it is also made of organic material, maybe it's not because it's relatively hard. Yeah, if it's also made of organic material, then hydrogen peroxide is also going to remove it. Yeah? So generally you add acid to remove calcium carbonate, and you add hydrogen peroxide to remove organic material. So if the mass stacks is made of inorganic material, then I think you should be able to use hydrogen peroxide. Yeah? If I make permanent slides, what should I record on the label to make it more professional? Professional under quotation marks. What I like to recommend, let's put it this way: when I see slides, especially antique slides that were made some long time ago, I find it very frustrating if there is no date on the slide, right? So you can estimate the time range when the slide was made, but I generally say the following: what it shows, and you can add slide labels on the left and the right side, right? Of course, what it shows, I would say the date, right? You can put your name or some kind of for branding purposes on it. Some companies do that as well. Possibly also the medium used for mounting it and stains. So what you could do is you could, let's say, use the left side of the slide. I don't even have, yeah, let's use this here. Okay. See, I cannot even separate this anymore. Okay. You could, if you have if you've got the specimen in the center, you could use, I don't know, the left or the right side over here. You could use as, saying what it is, larger text. And on the other side, I would probably include the date, possibly some processing techniques, mounting medium use, what stains you used, whatever you consider important. What I also have done is I've also sometimes used not only a date, but also next to the date a running number. So it's kind of like an index number. So if you want to keep track of it. So I've even, I've seen so many different slides with all different types of how do you say, labels on it. But I would say the most interesting thing, especially if the slides are supposed to survive for a longer time, would be the date. And unfortunately, I have so far not seen any antique slides with a date. It's a little bit of a pity. Instead of hydrogen peroxide, perhaps a differential centrifugation. Also possible, but if you're referring now to the diatom extraction, the idea of adding hydrogen peroxide also to diatoms is because you want to remove the organic content and you just want to have the nice clean shell of the diatom. Yeah, so this is actually one of the the reasons, because some people would like to just prepare the diatom shells, the silica shells, because without contents, without the cell's contents, sometimes the shells look actually very nice because it's just a shell, right? And there's no disturbing contents in there that kind of could distract from the pattern. Okay, so this is just something that I would like to share with you. I hope that I kind of addressed all of your questions in a satisfactory way. Let me quickly go through the couple of points which I might not have covered yet. Okay, let me just see. Okay, there are a couple of questions which are not on the sheet, which I'm also going to answer now. Differential centrifuge. Yes, here we go. Which type of filter do you have? How do you make dark field filter for clip-in filters? Should the width of these filters be 2 to 3 millimeters like the slide-ins, or about 4 millimeters for either plug out? So basically, what I have is I've got so-called prisms in there because my microscope has a technique called DIC. So I'm not using any color filters or anything like that. However, with color filters and so on, you can get a lot of, variations as well. How do you make dark field filters? Please do check my other channel. I have another channel called Microbehunter Microscopy, which is a little bit more technical, and where I've published several videos on how to make filters. Sometimes I'm actually going into a little bit more detail, which also answers a little bit the question about the size of the filters. You have to use a little bit of experimentation, okay? Generally the size of the filters, of the patch stop in dark field it can be larger than a certain critical value, but it should not be smaller, right? And finding this critical value is something I'm also explaining in the video. So it's called Microbehunter Microscopy. There should be a link anywhere somewhere. And it's my second microscopy channel. I've not been recently publishing so many videos on that one, but this is where I actually talked more about filters. Yeah. I'm trying to ask this strange biological question, but since urine is sterile, how come does it smell so bad after a few days? That's an easy to answer question, because it's only sterile at the beginning. Okay. Usually what happens, because urine does contain urea, which is an organic substance, and also a whole bunch of other nutrients. Bacteria will start to grow in the urine as soon as possible, right? So that is the reason, right? It becomes spoiled essentially. Okay, so that's the short answer. That's why, indeed, you're correct that fresh urine should be ideally be free of bacteria. Okay, no questions. Again, thank you very much to Curtis for the donation, and of course, I also want to say thank you to Scott for the very generous donation, to Rafael Lorenzo Alonso for the donation, also to Michelle for the donation. Thank you very much. Okay. Again, as I said, I'm going to use this money, of course, to buy again microscope slides, and I would like to show them to you. Yeah? Let me quickly check again. No, there is another one. This is probably a very basic question, but I get confused when you use the term depth of field. I have a feeling of what you mean, but I'm not sure. Could you elaborate this? Why not? Because it's actually a central topic. I need to, give you, I don't know which one. I'm just going to use the one that I have over here. Let's do the following. Okay. So if you just look at this here, then you're going to see that pretty much everything is in focus, right? Basically, it doesn't matter if the algae are on top or on the bottom, they're all basically in focus. So that's the depth of field, and the reason is because this objective here, the low power, has a large depth of field. So everything that's basically within this range, it doesn't matter if it's further on the top or further on the bottom, is basically in focus. Now look what happens if I go up with the magnification. Let's really go up here. Okay. And I need to find again see, I went up too quickly. Not a good idea. You should never change magnification too quickly, because then you're going to lose the focus. So here we go. Okay, let's use this here. So, and look at this here right now. You're now going to see that, let's say that this one here on top, yeah, is in focus, but the one over here is blurry because it's further down or further up. And then the reason is because the depth of field, that's kind of the area where something is in focus, is smaller, right? So everything that's outside of this slice, so to say, becomes blurry. So if you want to see something, yeah, very large, very thick, like for example the clam that we had, right? You're going to see that the part of the clam was always blurry, right? Because it was outside of this range, this depth of field, right? And so when you use microscopes, what you do is you've got this condenser, and when I close the condenser, what happens is it not only becomes darker, but also the depth of field increases. So now you're able to see that the things that are in the back are a little bit more in focus than before. And look what happens when I open it up, it becomes brighter. So I'm going to go down, yeah. And you see now the depth of field is lower because now it's very blurry right now. Huh? So that's basically the thing that I refer to when calling depth of field. So if there's some parts that are blurry, then it means that they're not in focus because they might be outside of the depth of field. Yeah? So that is the thing. By the way, it's not only for microscopes, but also for cameras, yeah, and so on. Yeah. So now I understand sterile means no bacteria. Yeah, not only no bacteria, but you could also say maybe no viruses or no living things in general, right? So that is, yeah. So I think I'm going to call it, quits for today. Again, I hope I did not overlook any questions. Ah, yes, there is one. Please recommend an English book for microscopy. That is difficult because it really depends what you would like to know, right? So, but I'm going to do the following. Maybe that's difficult. There are several books that Amazon, right? However, I do have to tell you that for whatever interesting reason, there are way more books in the German language about microscopy than in English, which is kind of interesting. So concerning hobby microscopy, amateur microscopy, microscope optics, and so on, much more literature available in the German language. Even identification books. It's kind of interesting, even though the English market should actually be much bigger. There are a couple of very nice English-speaking microscopy books, but sometimes they're not very practical. Okay, so they're generally about microscopy or about the microscopic world, electron microscopy, even beautiful pictures, right? But not so much hands-on. Or not so much biology even, right? I can show you. You see, the problem that I have is I'm a little bit worried because I would like to actually show you the contents of the books, but I'm kind of always a little bit worried about these copyright things, So what I could do is I could show you the cover of the books, but not the inside. What I do recommend is that for some of the books, there is on Amazon, you can actually look at a couple of pages in the book. So sometimes there's this preview available. So I recommend that you use that because that's obviously not a problem. But I would like to, simply say that it depends a lot on what your expectations are. And however, there are these days a lot of resources also available online. So this is also something that I would like to recommend. I'm going to call it now I'm going to call it quits for today again. I wish you all the best. Happy microbe hunting. I'm always, how do you say this, welcoming suggestions for things to put under the microscope, obviously. Leave your comments. And if you have not subscribed, I think it would be very nice. Also, I know that it's kind of a little bit awkward always click the subscribe button and like the video and so on. But not only mine, but generally, if you also subscribe and like other people who have microscopy channels, then this also kind of promotes the hobby a little bit, right? And it kind of gives a little motivational boost also to other people who have a microscopy channel. And there are several out there, right? So, bye-bye. I think enough for today. it's also time for me to go to bed now. Not everywhere in the world. Yeah, I know that some of you have also have been joining at very inconvenient times. I would like to thank you for that as well. But for now, all the best. Happy microbe hunting as always. See you around next weekend, and bye bye.